Approximately 350 million tons of plastics are produced globally each year, resulting in over 5.25 trillion pieces of macro and microplastics currently in the ocean. Microplastics have been reported to infiltrate various consumer products, including beer, salt, and drinking water, further exacerbating human exposure. The widespread accumulation of plastic pollution has had devastating effects on marine ecosystems. Ocean animals suffer from the ingestion and entanglement in plastic debris, leading to fatalities due to suffocation, malnutrition, and poisoning. To solve this issue, researchers have begun to turn towards a resource found readily available everywhere: algae. Algae has a well-characterized life cycle. It grows readily in commercial settings and has been demonstrated to make high levels of protein. These qualities make it an ideal organism for large-scale cultivations in a variety of applications. In our project, we are using Chlorella Cavaliers to produce large quantities of plastic-degrading enzymes. Previous studies have demonstrated that chlorella attaches to and colonizes plastic surfaces and can partially degrade type 1 plastics. With these key characteristics, we developed a project to help use algae to improve ecosystems through the degradation of plastics and hopefully generate high-value biomass.
In order to produce a strain of the Chlorella vulgaris that would produce our metase-petase enzyme complex, we utilized the central dogma in biology. The central dogma explains that DNA makes RNA, which in turn makes protein. We first started with our DNA. We created a plasmid that contained a cauliflower mosaic virus promoter that is used in most algae. We also added a codon-optimized metase-petase gene sequence and a hygromycin resistance gene that we would then use to select our algae. From this, we created a second plasmid, which included all the same components. However, on the N-terminus, we added a small section that would help to produce our enzymes or secrete our enzymes. We called this our secPep vector. Our goal was to then take these two plasmids and introduce them to our chlorella so that hopefully our chlorella would produce the METPET enzyme complex and secrete them into our environment.
Once we received our custom DNA, we came up with two methods to get it into our algae. The first method was electroporation. For electroporation, we first purified our DNA sample, and then we used electricity to open up our algae cell. Our second method was agrobacterium. Well, agrobacterium is a pathogen that infects algae and plants and has been known to introduce DNA sequences into plant and algae cells. For our project, we transferred agrobacterium with our plasmid. We co-cultured our chlorella with the bacterium, and then after the co-culture, we used antibiotics to kill any remaining bacteria, leaving only our algae.
After introducing metase and petase into our plasmid, we transformed the clones, screened the clones using hygromycin. The algae clones that did not have our hygromycin resistant gene did not grow. So that way we knew that we had our plasmid with the metase and petase gene. After several weeks of growth, we screened and collected more clones and tested them using PCR for our metase and petase plasmid. To test for plastic degradation, we used a recycled PET bottle from a soda bottle, and we pre-rated it and grew it in our algae plasmid for about a week to test for any degradation.
In the lab, we successfully developed, for the first time ever, multiple clones of chlorella containing our DNA vectors. In plastic degradation assays, we found that our cloned strains of chlorella degraded more plastic than wild-type or naturally occurring Chlorella vulgaris. We hope that soon our strains of algae can be used to efficiently degrade type 1 plastics from the environment and, at the same time, produce valuable algal biomass.
After we discovered our clones capable of degrading plastic, we went out into our environment to find some PET plastic to test. We gathered plastic and trash from a local stream, making sure to keep any PET plastics for further research, and we disposed of the rest properly. We hope that this can inspire others to go out into their environments and keep our waters clean.